Right, thank you very much. Morning, everybody. I've realised I don't have my video on for the Zoom, so I shall start.
No, that's fine. You don't need
to
put it
I
on.
don't need.
Martin's covering now.
Oh, grand job. All right. Cool. Okay, so yeah, I'm here to talk about data visualization in publishing and communication, which is a particularly awkwardly framed title, but never mind. Yeah, some of the things I'm saying and I will be saying you will have heard before, probably repeatedly and definitely yesterday, but a bit of a revision doesn't necessarily hurt. So, come on. Move on the screen. There we go. So fundamentally, let's get down to basics. Why do we bother with scientific communication in the first place, right? And there's this famous quote by Isaac Newton about standing on the shoulders of giants. Basically, science progresses as a whole by developing what is known and finding out what isn't known, or finding answers to what isn't known. Basically, you don't have to be a giant to support someone else's research. You just have to be able to communicate yours effectively. And that is, so rather than having the notion of a giant and then you just standing on their shoulders, what we actually have is this massive pyramid of people, or trapezoid of people, massive block of people, and you are kind of building on their work in a step-by-step initiative basis to increase the global knowledge that we already have. So, question then is, why do we, I mean, there's lots of ways we can communicate and disseminate our work. Why do we bother writing articles? Basically, articles are a good way of telling somebody about your research where you're not actually having to be in the same room as them, and they can go and read the article, and they can be in a different country, in a different time zone. They can be reading it many years after the event, and it's an asynchronous way of communicating research. And of course, one of the benefits of publishing a work in a peer-reviewed journal is the fact that you do get peer review happening. That is not only a kind of a quality control and check for you and your research work, but it also gives the readers of your article that bit of confirmation and comfort, knowing that the fact that, okay, this stuff has been looked at, it has been checked. It is, in as much as any bit of science is ever completely, true and verified, it is as true and verified as we can possibly be given our understanding of the time. I get often asked questions, why do journals care about data? And again, this is coming very much back to fundamental principles of how and why we do science. There's the difference between science and alchemy in that science is reproducible and you share your research. Historically, alchemists learned lots of cool stuff about chemistry, but they never shared their research with anybody. They never told what they were doing because they were all fixated on the Philosopher's Stone and turning lead into gold and all the rest of it, which meant that chemistry as a whole didn't really start becoming a science until people started sharing their results with each other and communicating with each other, and then going, oh, this particular method failed for me, so that means that you don't have to go and try it because it obviously doesn't work. So yeah, science should be reproducible. Other people doing the same experiments in the same way should get the same results. Now, of course, there's different domains have different kind of complications with this because there are some domains where you cannot reproduce the data. For example, if I went out, if I wanted to take measurements of how sunny it was yesterday, I can't do that today. I don't have a time machine. Observational data is not reproducible. So if you have the situation where you can't go and reproduce the data, you need to have access to the data that was actually collected at the time of the experiments, and you need to have access to that to confirm the science is valid, and of course check that that what the conclusions are being drawn from the data are actually plausible and confirmable. And of course, poor data analysis gives us false facts and inaccessible data cause all sorts of problems, and they undermine science and the credibility of science. And we don't want that. We want things to be, we want our science to be reproducible and verifiable. everybody throughout history has struggled with how to communicate data, how to visualize data. And these are some really old examples of sharing research data and visualizing it there as well. In that we've got the suber cells and mimosa leaves from Robert Hooke's Micrographia back 1665. That's pretty much one of the very first scientific figures there. And then we've got the table, which is also a valid method of data visualization, although people tend to kind of neglect it nowadays in favor of fancy interactive gubbins. But then tables are, as you can see, there's not that much data in there, but that data was very, very carefully collected and looked after. And it's from the Observations of Stars in the Spiral Nebula by William Parsons, the 3rd Earl of Ross, back in the 1800s. Of course, these are old data sets. Come on. Next slide. There we go. Data sets have gotten a lot bigger. I love this picture. Unfortunately, the Wellcome Trust don't have this on display anymore, but this is a hard copy of the human genome. That bookcase is taller than I am, and the books are very heavy, and they are written in very, very small writing. This is a way of visualizing the data in the human genome. It's not a very helpful way of visualizing the data in the human genome, but it's still kind of cool if you want to impress on people just the vast quantities of data and how much information there is. If you want to use this visualization for helping people understanding what a certain gene does, no, that's not the way to do it at all. The other thing that I'm always constantly aware of is that creating, analyzing, and describing data is hard work, and visualizing it is also hard work. I mean, we've got lots of cool tools and services to enable us to do data visualization, which is brilliant. But there is still an awful lot of mental effort that goes into how you convert from the massive data set that you've collected into what is effectively one graph. And of course, you're throwing out, or no, not throwing out necessarily, but you are synthesizing and pulling stuff together and highlighting various aspects of the data set that you have collected in order to be able to present it in a certain view. It's a way of taking slices through. All right, example, imagine you've got a 3D scan of something. You can either display the 3D scan as a video of two-dimensional slices, or you can just take a two-dimensional slice and look at that individually. When you are looking and thinking of how you're doing your data visualization, you have to think of, okay, what aspects am I looking at? What 2D slice am I taking out of my 3D data set? Okay. But it's hard work, so don't underestimate that. This slide I borrowed from my friend Quincy Justman, who was editor-in-chief of Cell Systems. And this slide is really interesting because it's showing, for the case study of biology, biology is changing very rapidly. So we have on the right, in figure A, we've got, and the two figures, A and the other one, so 1998 and 2018. 20 years apart, same journal, same laboratory, publishing research, and look at the difference between the figures. I mean, you've got on the left-hand side, you just have a plain two-dimensional plot. Looks like a cumulative distribution function there, but I could be wrong. And there's three graphs. There's three lines on that. If you look at the one on the right, there are 1, 2, 3, 4, 5 separate sub-panels, each of which, so there's that, each of which contains as much, if not more, information than the one on the earlier one did. And it's they've all got different ways of visualizing things. So you've got graphs and schematic diagrams and things like that. We have, and this is an example of biology, but it also is occurring throughout all the other domains as well. We are having more data, we're having better ways of visualizing that data, but they are making things an awful lot more complicated. So Quick kind of pop quiz for people to see if anyone's awake this morning. Does anyone, so given that example raw data set, does anyone want to hazard a guess as to what it actually is? You've got it. Yep, it's a normal distribution, rolls of 2 dice. Well done. That's, yeah. So basically, what the example there was, people were having a look at the raw data and it's kind of like, it's a load of numbers. But then when you plot them, it makes it so much easier to detect patterns. There we go. That's why we do it. Humans have evolved to be able to spot visual patterns very easily. Sometimes our ability to spot visual patterns is so easy that we do it on random noise, which is also an issue that you have to be aware of. Plotting things and putting data visualization is a really handy way for us to go, oh yes, that is a normal distribution, and there is the underlying kind of patterns in the data. The reason the example raw data is has different patterns in is because I started off kind of going, oh, how do I convert that particular graph into an array? And I just went bam and kind of went forwards and back along, starting from the top and backwards and working down. So there we go. But yeah, so. You've heard that pictures tells 1,000 words. Some pictures tell far more than that. So this is the Human Impacts Database, and the web link is given there. It is a collection of useful numbers for quantifying the impacts of the human presence on Earth. So basically, you go to the website, and you can type in livestock population or emissions or ocean pH or whatever, and it will pull up data which is fully referenced and described as, and so you can find out all sorts of interesting numbers, right? The image on the right-hand side, however, it comes from the author's bioarchive paper, and we're also in the process of, so it comes from their bioarchive paper, and as you can see, the fact that it's an infographic, it's got a lot of numbers involved in there, but it is presented in a way so that it's visually appealing, even though it's quite small. You can look at various things are grouped according to different things. So you've got your city there, and you've got your material production, and you've got your human population around your city. Then you've got your coal-powered power station there, and you've got your greenhouse gas emissions and power from fossil fuels and total power use and things like that as well. So it's a really nice visual way of having people let their eye being caught by say the farm, and then you can look down a bit closer and go, oh right, there's an awful lot of chickens. There's about 4 chickens per human at the moment in the world, and there's and for cattle, there's about 1 cow per 5 humans. Same for pigs. All livestock, there's about 6 animals per human in this world, which is kind of cool when you think about it. So anyway, so that infographic is very well designed, very well put together, and it looks really cool. So, so I recommend having a think about that. And this is all about how you tell a story with your data sets as well, or with your data visualization. So I could not do a talk without doing an errant science picture, and this one came out yesterday, which was absolutely perfect timing. And this is how you use data visualization to tell a story, right? So here we go. We have the average year in research. And we've got our nice little bar chart, and it's broken down total hours in the year, official work hours, holiday time, which is sleeping, and then how many hours it feels like I worked, and you can see that the bar for that kind of goes off the charts completely. And then it's then how hours I actually worked, which is that. And then, of course, then because what you're actually interested in is the hours I actually worked, that gets pulled out and split up into meetings, preparing for meetings, fixing lab equipment, coffee, actual research time and writing. And then, of course, the writing time gets pulled out to into that pie chart, which, it's a different way of producing information or showing information, but you've got your papers, books, emails, staring blankly at the screen, which is definitely what happens when you're attempting to write stuff, and then the Google search bar, and then out we go in how do I recover Word document after crash? How do I spell something or other, and cats falling over, definitely the internet is meant for cats. And then also 2% of the time, how do you calculate percentages? Because, yeah. So this is a really nice infographic that takes you very smoothly and easily through the story of what the artist is doing with their day and with their time. And it's really cute. I really like it. And it's very, very fun as well. Sammy mentioned that we do graphical abstracts in patterns, and yes, we do. They're not mandatory, but it is quite handy to have them. Here is a graphical abstract from this paper, Deep DRID, Diabetic Retinopathy. So it was a grading and image quality estimation challenge, so it was a data science challenge. And here we've got another example of what is effectively using data visualization and graphics to tell a story about what was going on. So we've got everything's nicely done in columns that follow one after the other. So you've got your data collection column, you've got your algorithm on site, and then rank, publication, and summarization. And within those things, it's fairly easy to go, okay, columns go left to right, information in the columns go from top to bottom. There we go, sorted. So it's fairly easy to just zip down and see what's happening, okay? This, I think, is a good example of a graphical abstract. This, yes, is not quite such a good example of a graphical abstract, okay? So the article was about machine learning approaches in COVID-19 survival analysis and discharge time likelihood prediction using clinical data. Okay, I mean, you do have bits of information in that graphical abstract. I don't want to kind of slam the authors too heavily for this one, but I think you can draw your own conclusions from that one. Yes, so moving swiftly onwards. Back in 2020, we also published an article called Principles of Effective Data Visualization. And these are kind of, again, generic things that people think about already, but it's handy to kind of have a bit of a reminder. When you're doing complicated graphical abstracts or flowcharts or whatever, get the piece of paper out and the pencil and diagram it first. Rough it out so you can have an understanding of where the flow goes and what the story you're telling is and how you lead people through that storytelling kind of path throughout the image. Using the right software, we've had a lot of discussion and work with the right software. Use an effective geometry and show your data. That's important too. Colors always mean something is something that I find to be really interesting. And you've also got to remember, yes, colors may always mean something, but not everyone can see all the colors that you put down there. So please, please, please be accessible. Include uncertainty. So you've got your error bars and things like that. And they are very important as well. If you are doing multiple graphs, if they are all related into the same basic thing panel, if you can, but don't have umpteen billion panels that run over multiple pages because that will just confuse people. Data and models are different things. Yes, they are indeed. Simple visuals, detailed captions. So yeah, you want people to be able to take a look and go, okay, I think I can understand that. Let's have a read of the caption. Ah, right, that explains that. Rather than having a look at it and going, I've no idea what I'm looking at, and then reading the caption and discovering that you still have no idea what you're looking at. You need to be clear about what it is you're displaying and why. Infographics we've already talked about, and then get an opinion. If you are unsure about whether or not your data visualization says what you think it says, ask a colleague, ask a friend, ask a family member, depending on family members, basically. I wouldn't advise asking the cat because the cats tend to not be that fussed by data visualization, but you can always explain things to your cat or your pet or your rubber duck or whatever, and talk through the flow of the image, because that's a really handy way of figuring out what's going wrong, not only in your code if you're having coding problems, but also kind of taking you through bit by bit on your data visualization and explaining what's going on. And then sometimes you can spot things that have gone a bit haywire in there as well. More general hints and tips. Beware of chart junk. Don't put stuff on there unless it's going to be useful. For the love of whoever, deity, please label your axes. That drives me spare. Yeah, make sure that when you're submitting article or submitting paper, make sure when you are submitting your figures attached to your paper that you include high quality versions of them so that people can zoom in without the resolution going haywire. And I've mentioned this before, if you're using color, brilliant, think about accessibility. Always think about accessibility, that's very important. Okay. I've mentioned it's okay to ask for help. People looking at something with fresh eyes are great for spotting elusive mistakes or places where things could be clearer or whatever. Peer reviewers, of course, do this as their job of peer review. But please, 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 again, make sure that you've done a good proofread before you submit your article and your images, because peer reviewers tend, understandably, to get a bit cranky if they keep running across spelling mistakes every couple of sentences. I get pretty cranky running across spelling mistakes every couple of sentences as well, and I have to read the papers first, so yeah. Okay, something that you may or may not be aware of, but some journals, my journal and my organization, we have this thing called Image Detective. So as well as having Authenticate, which is a software designed to check for plagiarism for stuff posted on the internet, we also have Image Detective, which checks for image modifications in figures. And basically, here we have an example of, so we've got the source on the left and the detected on the right. And basically, Image Detective is complaining because it says marked region inadequate image quality, saying low resolution or high compression. Now, this is something that got flagged in one of the articles that we published, and I looked at it and went, yes, that's all right, because what the source is showing the original image and then the various different model results as a result from those original images. So the fact that there was low resolution or high compression in there, it wasn't really so much of a problem for us. And the authors did provide a higher resolution image later on anyway. But that is something that there is software around that can do image detection and can pull out low resolution things. It can pull out areas where things have been cropped, where things have been copied and rotated and pasted again. So there are ways of checking images for manipulation. Whether that's intentional or unintentional is up for debate either. Of course, the brave new world that we're living in, we can get AIs to generate images for us. So this is Dali Mini, which is a really cute little fun image generating, I think it's a GAN, a neural network anyway. And I put in scientific graphs, and that's the sort of stuff we got, which seems to suggest that scientific graphs are mainly nodes and networks and stuff like that. But they look cute. They're completely inaccurate and all the rest of it. And you take one look and go, nah, that's not real science. That's not real science at all. The problem that happens, though, is that the same technology that can be used to create dream graphs or fake graphs like we saw on the last slide can also be used to generate images for the fraud in scientific purposes. We've published an article on this as well fairly recently. And it talks about, it's a perspective about the potential image fraud based on AI generative technologies. And the article talks about the scenarios, capabilities, and effects of AI algorithms that could be used for academic fraud. And that's something we as editors and you as peer reviewers and editors as well need to bear in mind. Now, I think I might be being a massive optimist here, but I think for the most part, most people will play fair and will provide the images correctly, and they won't be messing around with AI-generated images or anything like that. But this technology is possible, and there are always some bad actors around there, so it's just worth keeping in mind. I'm going to quickly, because I've only got a few more minutes, I'm going to quickly zip through my last few bits of slides. They're again, more generic, talking about data availability statements. It's data and code availability statements as well. And basically, we very much encourage you to write data and code availability statements telling us where your data and code are, and making sure that the data and code are available in a trusted repository, preferably with a DOI, because that makes life easier for your research, for your readers, and for people who want to build on your research afterwards. There is this thing that has been happening historically of lead authors going, data will be made available upon reasonable request. Yeah, that drives me somewhat spare as well. Because the question is, what does reasonable request actually mean? Does the request have to come with a bottle of 20-year-old whiskey attached with it? Or is it just, please, can I have your data? Thank you very much. And then, of course, there's not very future-proof either. What happens 20 years from now when the lead author has moved on, no longer has access to the institutional servers, or the CDs have turned into a giant melted mass in a cupboard somewhere? How do people deal with that? So we definitely encourage researchers to make their data available and fair, findable, accessible, interoperable, while acknowledging the fact that it is really hard work for people to do this. And it's really hard work for people to do data visualization as well. So as open as possible, as closed as necessary. This is the same with data visualizations too. It's some data can't be made open to everybody to use, because of confidentiality issues, conservation issues, security issues, all sorts of fun stuff like that. You also need to think in your data visualizations, if you are dealing with sensitive data, how detailed are your visualizations? Is it going to be possible for somebody looking at a visualization to uniquely identify one person in that particular in your particular thing, especially if they're an outlier in your graph or your point plot or whatever? It's very important to think about these things as well. And that is where I will finish up. So thank you very much.